presentation deals about the effect of different light conditions on the survival and growth of paralarvae of Octopus vulgaris under culture. Well, in our facilities in Tenerife, in the Canary Islands, we are working trying to culture Octopus vulgaris, focusing on the paralarvae stage, and there is massive paralarval mortalities during the first 20-30 days. So, probably due to nutritional or also zootechnical problems associated with stress problem. For this reason, light could be one of the main factors, is our hypothesis. Because this animal, you know, it has a sophisticated visual system, they can polarization, richness, okay, is very complex. Previous communication has talked about this. But there is no enough information. There is a very limited information about this, the light effect on the paralarvae. Regarding this, in wild population, one of the authors, Roura, and other colleagues, has studied the paralarvae in the natural environment. So they found, or they suggest, the data suggests that there is a vertical migration, this in the open ocean, okay? During the night, you can find paralarvae near of the surface, but during the day, you only found paralarvae at 100, 200, or even 500 meters. So this implies changes of the colors, blue, predominance of the blue colors in the depth, or intensity, etc. You must be taken into account that, for example, until now, in the culture, under culture condition, we usually we use 1,000 lux or 500 lux, but 100 meters maybe are 10 lux. So our question is, well, maybe we are using too much light intensity or not the correct color. Well, this is the hypothesis, no? Also, in addition, the paralarvae has it's a complex behavior because the first paralarvae, during the first days, until 15-20 days, they are near of the coast, but all the paralarvae are only found in the open ocean. Okay? Unless it is in a study, this study has been made in Vigo, coastal northwest Spain, and also in the Morocco coast. In both places there are upwelling. These data are focused in these places. No? I don't know data from other places. So we use this data. The objectives was to make several light assays and to test ecological-driven hypotheses, comparing the effects on growth and survival. Well, methodology. We use paralarvae for 20 days old, or 20 days old paralarvae. We use this condition, LED or fluorescent light, depending on the experiment, using light filter, renovation, typical condition of culture, called paralarval culture. We feed with artemia for nauplio for the first days and metanoplio for the more older paralarvae. The best diet is crustacean soil, but it's not possible because it's very difficult to obtain enough quantity to make a study like this with many replicate higher volumes. So we use 100 liter tanks in order to get enough quantity of replication or to test, to compare different treatments. 24 tons You can see here, this is a picture of this tank, all with the same condition. Using this kind of filter, 500 micrometers. And well, we use the data treatment was with a multifactorial ANOVA. Well, the first experiment were with LED light. We make this experiment. We use this row was with normal light. In this tank, we use a polarized filter. Okay. In addition, we use two different intensity, 10 lux and 80 lux. Why? Because we compare with oceanographic data, and 10 lux is more similar to 100 meters in depth, but 80 lux is near of the surface, 5 meters maybe, depend of the condition, is height will vary. There is a height variation. So, to condition blue light and white light. I am talking about lux, but lux is not the best way to take measure of the intensity, because the luxometer is the most the typical device for this purpose in the culture facility, but because it's cheap, it's very cheap. You need, but for a correct measure, you need a radiometer. Radiometer is in this case, we use a radiometer to compare the measure, because the radiometer, the measure is like flux, is the correct way of expression of the light intensity. In this case, you can say that the blue light has only one peak with this value, and the white light is a blue peak, green peak, and red peak. Okay, because this is the typical spectrum of a LED light. It's three different LED that together get a white light. Well, in this case, I am going to talk about lux in the next slide, but always you can maintain in mind that this is the correct value. Well, okay. So, when we compare the survival, in this case, under this condition, you can see this is the percentage of survival after 20 days. So, normal light or polarized light, different intensity or different colors. Well, when we use the multifactorial ANOVA, you can see a significant positive effect of color and no polarized light, blue and no polarized light, because no factor was found significant difference. Were found. No difference in intensity, no difference in color, no difference in polarized. But yes, there was a significant interaction. And when we compare the gray white, no difference. So you can see that there is a height variability and lower survival, but it's a normal survival at this stage, 20 days, sorry. Well, according to this data, we designed a second experiment, also with LED light, but in this case, we only use normal light. We use three intensities. The same intensity, 10, 80, and 270 lux. Two colors, the same colors, and the same intensities. The same intensity, I mean, the same spectrum. So the result, when we take measure of survival, we found these values, but there is no significant difference. No interaction, no difference in intensity, no difference in light. But when we take measure of dry weight at 20 days old, we found tendencies that there is a significant difference in the dry weight. There is a lower dry weight at lower intensity, 80 and 10 lux. This really, we don't expect this result because this intensities is the intensity of 100 meters. So we thought that maybe the paralarvae was better than the low light intensity. But no, it's not the case. There is not a good growth. So for obtain a good growth, we need to increase the intensity. According to this, we designed a third experiment, but in this case we have to use fluorescent light. Why? Because we want to increase the intensity and we cannot increase the intensity with this LED light, not with our LED lights. No? So we increase the intensity to 300 and 600 lux and normal light. But also, in this case, we obtain the blue and white with oceanographic filters. Of course, this changed, so we cannot compare with our previous experiment, but we cannot take other option. In this experiment, we include a different factor, a new factor. We cover the tanks with the same condition, but we half covered with a mesh. You can see in the picture here, we put a mesh in the middle of the tank. Asking, well, what matter if the paralarvae can choose, if they can be under the light or they can in a different, in a subtle zone. Before the result, I'm going to explain what is the light profile of this experiment. When you use fluorescent light, it's not like LED light, not only 3 peaks. There is a spectrum, many peaks with different in different wavelengths, and there's typical the fluorescent. And for example, here you can see what's matter when you use the different intensity of filter. Oceanographic filter, blue. You can see here, for example, the oceanographic filter increase the blue light here. When you compare with white, for example, no? In this case. But the white light, without filter, increase the other colors, for example here, like the yellow color, okay? Or also in the red color. Well, we also compare what's matter with in this experiment, we compare what matter with the light under the water. Just in this case, it's only 100 liters tank, a small tank. But, okay, you can see a small difference between surface and 30 centimeters. You can see that in blue light there is not too much difference, but in yellow or red there is some, in 30 centimeters, there is a lower level of yellow or red light, okay? It's normal, but curiously, only in 30 centimeters. So for future study, we are using bigger tanks, so we hope that the effect was bigger. For example, in these pictures, you can see the effects of comparing the intensity, how the intensity is reduced between surface and 30 centimeters. The blue light maintained only maybe 20%, but the yellow or red light reduce in bigger scale, no? So this is the effect. So taking into account this, the result about the survival was that the survival increased in the tank using saddle zone. Okay? In fact, we found a significant effect of the saddle zone in the half-covered tanks in respect to normal tank. So anyway, well, the conclusion is, okay, we improve the survival, but mainly due to this treatment. 600 looks, blue, and half-covered tanks. So there is an interaction, but also there is a significant effect of the saddle zone, just for this factor alone. Anyway, if you can see this result, there is a height variability. In this case, there is not too much. So we consider this experiment, this data, as initial data. We need to do more experiment to understand this interaction or to confirm this under other conditions, different conditions. So there is no difference in dry weight here. In conclusion, these results suggest that the light conditions are critical for the paralarvae. We know that low intensity are not good for the growth of the paralarvae, at least until 20 days old paralarvae. The half-covered tanks improve the survival, okay, but depend of the other condition. And there are several interactions. For this reason, I consider as initial data, no strong conclusion. In consequence, we think that we need to do more experiments to clarify this effect. But taking into account the type of light, intensity, spectrum, previous studies is very difficult to compile because the information is very scarce. There is no information, they don't take into account many aspects. The fluorescent light are very different from the LED light in many aspects. Also the environmental factor, the tank color, there is a strong influence, volume, the depth, the tank. Green water is very usual effect in the culture of the paralarvae. But also, a very important is the developmental stage of the paralarvae. Okay, we are working with 20 days old paralarvae, but feeding with Artemia. If you feed with Artemia paralarvae, the growth is very small compared with if you fed with crustacean soil. So, if the paralarvae change, the development of the paralarvae probably is going to be very different between the Artemia or crustacean soil. Maybe if we use crustacean soil, the result could be different. Okay, it's very complicated to do experiment with this. So we consider this like a first step. We would like to do experiment with paralarvae feeding with soil, with crustacean soil, like well.